Pharmapal.
Drug classification and actions in medicinal chemistry. Hey everyone! Welcome back to our channel. Today, we're diving into the fascinating world of medicinal chemistry, specifically focusing on drug classification. Whether you're a student, a professional, or just curious about how drugs are categorized, you're in the right place. So, what exactly is drug classification? In the realm of medicinal chemistry, drugs are categorized based on their medicinal uses rather than their chemical structure or origin. This approach helps us understand how different drugs work in the body and how they can be used to treat various conditions. Let's break it down into two main classes: pharmacodynamic agents and chemotherapeutic agents. First up, we have pharmacodynamic agents. These are drugs that interact with the body's physiological functions. Think of them as the ones that help manage symptoms or alter bodily processes. Examples include general anesthetics, which help you sleep during surgery, hypnotics and sedatives that calm anxiety, and analgesics that relieve pain. Now, let's talk about chemotherapeutic agents. These are the drugs that fight against pathogens, such as bacteria, viruses, and even cancer cells. This category includes antibiotics, antiviral medications, and anti-cancer drugs. They're like the body's little warriors, targeting and eliminating harmful invaders. Drugs can also be classified based on the types of diseases they treat. We can categorize them into three main groups. 1. Infectious diseases. These are caused by pathogens like bacteria, viruses, fungi, and parasites. Think pneumonia, AIDS, or malaria. 2. Non-infectious diseases. These are disorders that arise from genetic factors, environmental influences, or lifestyle choices. Conditions like diabetes, heart disease, and arthritis fall under this category. 3. Non-diseases. This is a bit different. It includes drugs that alleviate pain, prevent pregnancy, or induce anesthesia. Pretty interesting, right? But how do we determine which drugs belong to which category? The classification often relates to the physicochemical properties of the drugs, such as solubility, acidity, and basicity. These properties influence how a drug behaves in the body, how it's absorbed, distributed, metabolized, and excreted. For instance, a drug's solubility can greatly affect its bioavailability, which is how much of the drug actually reaches the bloodstream. If a drug is poorly soluble, it might not work effectively, no matter how good it is at targeting a disease. Additionally, the interaction of drug molecules with biological targets like proteins, enzymes, or even DNA plays a crucial role in their classification. Drugs can be either structurally specific or nonspecific in their action. Structurally specific drugs interact with specific receptors, while nonspecific drugs may affect multiple targets. Now, let's not forget about the importance of structure. The chemical structure of a drug can influence its activity, potency, and side effects. This is where concepts like stereochemistry and isosterism come into play. Stereochemistry refers to the 3D arrangement of atoms in a molecule, and it can significantly impact how a drug interacts with its target. For example, two enantiomers, molecules that are mirror images of each other, can have vastly different effects in the body. And there you have it. Drug classification in medicinal chemistry is a complex but crucial aspect of understanding how we develop and use medications. By categorizing drugs based on their uses, we can better appreciate their roles in treating various diseases and conditions. Physicochemical properties in relation to biological action. What we mean by physicochemical properties. These are the physical and chemical characteristics of a substance that influence how it behaves in a biological system. Think of them as the personality traits of a drug, how they interact with the body and how effective they can be. The three key properties we'll focus on today are solubility, acidity and basicity, and reactivity. Each of these plays a crucial role in how a drug functions once it's in the body. Let's start with solubility. Solubility is all about how well a drug can dissolve in different environments, like water or fats. Imagine trying to mix oil and water. They don't blend well, right? Similarly, a drug needs to be soluble in bodily fluids to be absorbed effectively. Now, drugs can be hygrophilic, which means they love water, or lipophilic, which means they love fats. Most effective drugs strike a balance between the two, allowing them to move through both water-based and fat-based environments in the body. This balance is crucial for absorption, distribution, metabolism, and excretion, often referred to as ADME. Next, let's talk about the intermolecular forces at play. The solubility of a drug is influenced by various types of bonding, like van der Waals forces, dipole-dipole interactions, and ionic bonding. Each of these forces can affect how a drug interacts with its environment and its target sites in the body. Moving on to acidity and basicity. Understanding whether a drug is an acid or a base is vital. Why? Because the ionization of a drug affects its solubility and permeability. For example, a weak acid like acetic acid has a specific pKa value that tells us how well it will ionize in different conditions, like in the stomach versus the intestines. When we talk about acids and bases, we're referring to their ability to donate or accept protons. This property can influence how a drug behaves in the body, including its absorption and how it interacts with receptors. Now, let's discuss reactivity. This property determines how a drug will chemically interact with other substances in the body. For instance, if a drug is highly reactive, it might break down too quickly, losing its effectiveness before it can do its job. Speaking of effectiveness, let's touch on drug receptor interactions. A drug needs to bind to its target receptor to elicit a pharmacological response. The fit between the drug and the receptor is influenced by the drug's physicochemical properties. If the drug structure doesn't match the receptor well, it might not work as intended. Hey there, welcome back to our channel. Today, we're diving deep into a fascinating topic in medicinal chemistry, the solubility of organic medicinal agents, or OMAs for short. Whether you're a student, a professional in the field, or just curious about how drugs work, this video is for you. So, let's get started. First off, why is solubility so important? Well, think of it this way, solubility plays a crucial role in how a drug is formulated and how it behaves in the body after it's taken. It affects everything from absorption to distribution, metabolism, and excretion. In simpler terms, if a drug is insoluble, it can't do its job effectively. Now, let's talk about solubility in terms of affinity. You might hear terms like hydrophilic and lipophilic thrown around a lot. So, what do these mean? Well, if a substance is hydrophilic, it's water-loving, it dissolves easily in water. On the flip side, lipophilic means lipid-loving, these substances prefer to dissolve in fats or oils. Most organic medicinal agents have a balanced solubility, meaning they can dissolve in both water and lipid environments. This balance is crucial because drugs need to move through both aqueous environments, like blood plasma, and lipid environments, like cell membranes. Now, let's visualize solubility as a continuum. On one end, we have highly lipophilic compounds, and on the other, highly hydrophilic ones. Most drugs fall somewhere in between. To dissolve in a solvent, a compound must establish attractive forces with the solvent molecules. Let's break down these forces. There are four main types of attractive forces involved in solubilization. 1. Van der Waals attraction. This is the weakest force, occurring between nonpolar groups and is highly dependent on distance and temperature. 2. Dipole-dipole bonding. This is stronger and occurs between molecules that have positive and negative ends, like water. Hydrogen bonding, which is a specific type of dipole interaction, is a key contributor to hydrophilicity. 3. Ionic bonding. This is a strong electrostatic attraction between positively and negatively charged ions. For ion-dipole bonding, this occurs between ions and polar molecules, like water, and is also quite strong. Now, how do we predict the solubility of an organic medicinal agent? Well, scientists often use a method called quantitative structure-activity relationship, or QSAR. This approach helps predict the solubility based on the chemical structure of the compound. Let's switch gears and talk about acidity and basicity. The acidic or basic properties of OMAs are crucial during both the formulation phase and when the drug interacts with its target in the body. An acid is a compound that can donate a proton, while a base accepts a proton. Common acidic groups include carboxylic acids and phenols, while basic groups include amines. The strength of these acids and bases can be estimated using their ionization constants, known as Ka values. A higher Ka means a stronger acid, while a lower Ka indicates a weaker acid. Next, let's touch on how solubility impacts drug design. When designing new drugs, chemists consider the balance of hydrophilic and lipophilic properties to ensure the drug can effectively reach its target site in the body. This balance is often quantified using the partition coefficient, which compares how well a drug dissolves in lipid versus aqueous environments. A higher partition coefficient indicates that a drug is more lipophilic, while a lower value suggests it's more hydrophilic. This information is crucial for predicting how a drug will behave in the body. To wrap things up, understanding the solubility of organic medicinal agents is essential for drug formulation and effectiveness. From the types of intermolecular forces to the role of acidity and basicity, every detail counts in the journey of a drug from the lab to the pharmacy shelf. Now, one of the key concepts is ionization. Most drugs are either weak acids or bases and can exist in ionized or unionized forms. Ionization is the process where a drug gains or loses a proton, resulting in charged molecules. This is crucial because the ionization state affects the drug's solubility and its ability to cross cell membranes. For instance, an ionized drug is usually more water-soluble, which is great for binding to receptors in the body. On the flip side, the unionized form is often more lipophilic, meaning it can easily pass through cell membranes. So, the balance between these two states is vital for effective drug absorption and action. The extent of ionization depends on the drug's pKa and the surrounding pH. For example, a drug with a pKa of 4 to 5 is considered an acidic drug. In a low pH environment, like the stomach, it may remain non-ionized, allowing for absorption. Conversely, in the higher pH of the intestine, it may become ionized and less absorbable. Next, let's discuss solubility. Solubility is the ability of a substance to dissolve in a solvent. It's crucial for two main reasons: the formulation of the drug into a usable dosage form and its biodisposition after administration. This includes absorption, distribution, metabolism, and excretion. The solubility of a drug can be influenced by various factors, including its chemical structure, the nature of the solvent, temperature, and pH. For example, polar functional groups like OH or NH2 enhance water solubility, while nonpolar hydrocarbon chains increase lipid solubility. Speaking of solubility, let's touch on hydrogen bonding. This special interaction occurs between a hydrogen atom attached to an electronegative atom and another electronegative atom. Hydrogen bonds can significantly affect the boiling point, melting point, and solubility of compounds. There are two types of hydrogen bonding: intermolecular, between different molecules, and intramolecular, within the same molecule. These interactions can stabilize certain structures and influence drug activity. Now, let's move on to complexation. This occurs when drugs bind to other molecules, forming complexes that can affect drug absorption. For instance, certain drugs may form non-absorbable complexes with substances like polyethylene glycol, which can reduce their effectiveness. The rate of absorption is directly related to the concentration of free drug molecules. When a drug binds to proteins in the blood, it forms a drug-protein complex, which acts as a reservoir. This binding can influence the drug's solubility, half-life, and interaction with other medications. Now, let's get into stereochemistry. This is the study of the three-dimensional arrangement of atoms in molecules. It's crucial because different stereoisomers can have vastly different pharmacological effects. For example, the enantiomers of a drug may bind differently to receptors, leading to variations in activity. Take warfarin, an anticoagulant. The S enantiomer is significantly more active than the R enantiomer. This stereospecificity is a key consideration in drug design and therapy. Lastly, let's talk about isosterism and bioisosterism. Isosters are compounds that have similar shapes and electronic properties, while bioisosters are groups that exhibit similar biological properties. This concept is widely used in drug design to modify compounds for better efficacy and reduced side effects. In conclusion, understanding these physicochemical properties helps us grasp how drugs work, how they can be optimized for better therapeutic effects, and the importance of their interactions within the body. Understanding acidity and basicity in chemistry. What exactly do we mean by acidity and basicity? In simple terms, acidity refers to the ability of a substance to donate protons, while basicity refers to the ability to accept protons. This is all based on the Bronsted-Lowry theory of acids and bases, which is one of the most widely accepted definitions in chemistry. So, an acid is any compound that can donate a hydrogen ion, or H+, to another substance. A classic example is hydrochloric acid, or HCl. When it dissolves in water, it donates a proton, making the solution acidic. On the flip side, a base is a compound that can accept a proton. Take sodium hydroxide, or NaOH, for instance. When it's mixed with water, it accepts protons, making the solution basic. Now, let's talk about something called the pKa value. This is a crucial concept in understanding the strength of acids and bases. The pKa is a numerical value that indicates how easily an acid can donate its proton. The lower the pKa, the stronger the acid. For example, acetic acid has a pKa of about 4.76, which tells us it's a weak acid compared to stronger acids like sulfuric acid, which has a pKa much lower than that. But what about bases? Well, we can also use a similar concept for bases, called pKb. Just like with pKa, a lower pKb means a stronger base. Now, you might be wondering, how do we determine the strength of these acids and bases? It all comes down to their ionization in water. When an acid dissolves in water, it breaks apart into ions. For instance, when acetic acid dissolves, it forms acetate ions and hydrogen ions. This ionization is what contributes to the acidity of the solution. The more ions produced, the stronger the acid. Similarly, for bases, the more hydroxide ions produced, the stronger the base. Speaking of ionization, let's touch on the concept of salt formation. When an acid reacts with a base, they neutralize each other, forming water and a salt. This is an important reaction in chemistry and can influence the solubility and bioavailability of drugs in medicinal chemistry. Now, let's consider the practical implications of acidity and basicity in our daily lives. For example, the pH scale ranges from 0 to 14, with 7 being neutral. Anything below 7 is acidic, and anything above 7 is basic. This scale is super important in fields like biology and environmental science, as it affects everything from soil health to human physiology. And here's a fun fact: our stomachs produce hydrochloric acid to help digest food. That's why it's crucial to maintain a balance in our body's pH levels. To sum it up, understanding acidity and basicity is essential for grasping many concepts in chemistry and biology. It helps us comprehend how substances interact, how drugs work, and even how our bodies function. If you have any questions or want to dive deeper into any specific area, feel free to drop a comment below. And don't forget to like and subscribe for more chemistry content. Thanks for watching, and until next time, stay curious.
Pharmapal.